Welcome to our three-part video series featuring experts from the Criterium Oncology Consortia. Hello, Dr. Messersmith. It's so nice to meet you.
Nice to meet you as well.
You have noted that today every cancer is a rare tumor due to its unique molecular characteristics, requiring teams of experts with experience and knowledge to tackle these distinct problems. How is the Academic GI Cancer Consortium, or the AGICC, meeting this challenge?
AGICC meets the challenge in several ways. So when I was beginning my career, we really just divided cancers up by where they came from. So in the GI world, we'd say, well, this is colon cancer, this is pancreas cancer, gastric cancer, what have you. And really didn't pay a lot of attention to the molecular characteristics, except out of sort of an interest of what might be coming, down the pike. And now we really can't take care of patients without fairly complete molecular testing. In fact, it's pretty much standard of care nowadays to check for 300, 400 different molecular markers, especially for widespread disease. And so you get these very complicated reports back, and you're trying to figure out what's the next best step for these patients. And it really requires two areas of expertise. One is expertise in the disease areas, because just because there's a gene X in a tumor doesn't mean necessarily that a therapy that targets that gene is going to necessarily work. So you really need to be familiar with the biology of these tumors, with what's coming out of the labs, what are they seeing, and cancer cell lines, and that kind of thing. And then the other area is what's going on in drug development. And one of the advantages of AGICC and the other consortia is that most of the members have early drug development expertise and experience, and you basically know what's coming down the pike a couple years before it hits the clinic. And so it really requires a merging of those things. One is the sort of the disease expertise, and then the other is the drug development expertise, and you're trying to basically match those things and develop trials which will help determine the new standards of care and time to come. That merging of expertise is really what makes the group unique, because these are people who only see GI cancers. I always tell people when I see them in clinic, look, there's over 200 different cancer types, but I only see about 6 or 7. I've only ever done that for about 20 years. And there's folks on the group that are even more experienced than me. And it's been great to see this transformation over the years, but it's gotten more and more and more specialized. We also work in multidisciplinary teams. So all of us who specialize in gastrointestinal cancer, for instance, we all work very closely with surgical oncologists who are specialized. They don't operate on anything. They operate really on GI cancers. You have very specialized hepatobiliary surgeons, for instance, who resect pancreas lesions or what have you. Radiation oncologists, pathologists, all the pathologists we work with are highly specialized. They really, focus only on GI cancers and nearly across the country. And we're also high volume, which is another thing. If you're looking for a needle in a haystack type of tumor, say a certain cancer with a certain biomarker, you need a big haystack. And so by merging over 20 academic centers, many of which also have satellites, here at the University of Colorado, for instance, we have a huge consortium. We have over 2 million patients in our system. But it's not only what we call the mothership, which is near Denver, but we also have very large hospitals north and south of us, and multiple satellite centers. So, just because it says one institution doesn't mean that you're actually not dealing with, 5 or 6 different hospitals and groups. And that's true of many of the consortium members as well. So it might list off 20 centers, but in fact, you might have more like 80 if you really think about all of the affiliates and satellites and all the other things.
With all that specialization, I would think that leads to some pretty cutting edge, different types of clinical trials. Can you talk about how this is changing, as you said, the drug development process?
Yeah, again, a lot of it comes down to basically being part of the drug development process where you have a sense of how trials are being developed, how they're being designed, ahead of time. And so while you're trying to make decisions about what a study might look like, or how to incorporate different groups or different tumor types, et cetera, it's just good to have a group of people where people know what's coming down the pike a few years before it's there. So you might have a conversation about designing a study in colorectal cancer, and you're in a group, room full of people who are saying, well, these are the results that are going to come out over the next couple of years. This is how these results might affect this trial design. So we should design the study so that it's relevant no matter what. Nothing's worse than putting all the work into a trial. And remember, these are patients we're talking about. I mean, I have to sit face to face with a patient and make sure I'm working on their behalf and putting them on the best, therapy that I can think of that's available at our site. And so we don't want to do a study and then find that it's irrelevant by the time it comes out. You really want to design things so that you know that through the life cycle of the study, it's still going to be important when it's said and done. And sometimes we'll even wait on study results. So we'll go ahead and have two different study designs that we're thinking about, but we want to wait for the trial results from X, and we know that it's coming out on July 12th or something. So we'll schedule a meeting a little bit later on so that we can react to it and then design our study accordingly. And so, in a sense, I sort of think about it as sort of legal insider information. In other words, all of us are on these confidentiality things, and we're all very careful about that. We don't discuss trial results that maybe we know about that are not publicly available. A lot of us are on data safety monitoring committees, and of course, we're very careful on all of these legal agreements and things that we need to pay attention to. But we are all aware, I shouldn't say all of us, everyone in the room has different areas that they might know about, so that we're kind of aware of what's coming later, and we can make sure we're designing studies that are going to be important 2 years from now. Because what's happening today really isn't all that relevant, honestly. You have to say, okay, what's going to be the standard in 2023? What what trial results are going to come out in 2022? How can we sort of, redesign this? I think one good example of this is in gastric cancer. Checkpoint inhibitors and immunotherapy were recently approved for the first line setting. And so that means in untreated patients, you're now using checkpoint inhibitors. Well, if you had a trial for later lines of therapy, so, okay, second line or third line, and we want patients who have never gotten a checkpoint inhibitor, well, your trial's dead. Those patients don't exist now. They're all going to be receiving it first line. Whereas if you think ahead and you say, there's trials going on, I'm pretty sure those trials are going to be positive. The phase 2 studies were positive. We should design studies that are looking for patients who have already been exposed to checkpoint inhibitors in gastric cancer. And so, we've been thinking about that since 2019 because we knew those studies were coming and that the results would be reporting out. That's the type of thing. It's kind of a specific example of where planning ahead and kind of knowing what's going on, across Europe. I mean, trials now are all international, right? So you need to know what's going on in Europe and Asia and the United States, and sort of be able to plan ahead on that.
And it's that specialization which makes it so unique in terms of what you all are doing, because it's the sharing of that data within the consortium that really is making a difference. Tell me how this personalized approach, and we keep hearing that cancer has now become a personalized disease, ultimately improves patient outcomes. So what can we expect as patients?
Yeah, so basically what's happened is, instead of saying somebody has, pancreas cancer, you're saying somebody has KRAS G12C mutated pancreas cancer. There's roughly 7 different KRAS mutations that we look out for, and we now have therapy specifically against G12C. which is a specific gene mutation. So basically, when we say that almost every cancer is a rare cancer now, what we mean is that the patients have now been sort of subsetted into these smaller groups, and you need to design a trial that's kind of personalized for that specific gene alteration, or a combination of gene alterations and things like that. So basically, you're putting patients into much smaller groups. You're using therapies that are targeted specifically to whatever gene mutations or alterations they might have in their tumor. That's totally basically changed the field. And again, to successfully conduct a trial in that setting, you need people who are familiar with the genetics. You need people who are testing. I still do a lot of second opinions where there's been zero testing sent. You need to make sure you have the test results, and then that you have the volumes and things that are needed to find all of those smaller, brackets of patients. At any given center, even a high volume center, you might only have a few. But if you have 20 centers, and again, a lot of them have satellites and other things, each of them has a few, well, pretty soon you're looking at 50 or 60 or 100, and you're able to actually get something done and move the field forward.
And that's the key, being able to tap into all those different centers, because as you said, you can't do it with 1 or 2 patients per center, but in a group, it's far more scalable and doable. And is that
exactly
right? And that's the beauty of the consortia.
Exactly. And the other thing is making sure people are doing state-of-the-art testing.
Hmm.
Sometimes, again, I'll do a second opinion from somebody who might be seen in the community or something, and they say, oh, I had molecular testing. They tested for 3 genes. It's like, well, we need to test for 400 genes. So, 3 genes isn't going to cut it. We're making sure that state-of-the-art testing has been done, and that, no stone has been left unturned in terms of trying to find if somebody has something that is potentially targetable.
And that molecular testing, is that a communications issue? How do we encourage everyone to undergo molecular testing to find out what their specific cancer is?
Yeah, it's a complicated factor. For a while, a lot of these tests were not FDA approved, and now they are.
Right, or covered
Um,
by insurance?
yeah, exactly. It still can be an issue making sure that it's covered by insurance, and some groups will have a relationship with a specific company. Obviously, I don't endorse like a specific company, but there's a couple of large companies that do this fairly in-depth testing, and the academic centers are all on board with that. And so, when I'm seeing someone who's been at another specialized place, they almost always have the state-of-the-art testing that we're looking for. But often people don't, and you can tell even with the folks that they're seeing that they're not quite sure how to interpret the test. So again, getting back to KRAS, I've seen clinical notes that say KRAS mutation positive. What does that mean? Which KRAS do they have? What did they test for? And so you have to sort of delve into that and try to get to the bottom of what did they actually send, what were the actual results, and make sure that's clearly documented. In some studies, we actually also do a central confirmation. So we make sure that the alteration we think they have is actually there. And increasingly, people are interested in liquid biopsies. So this is a blood test where you're actually looking for tiny pieces of mutated DNA that really shouldn't be there if you don't have cancer. And of course, the vast majority of DNA circulating in our systems is normal. If you don't have cancer, you have DNA circulating in your blood. But what we're looking for is gene mutations, methylation patterns, other epigenetic abnormalities that really shouldn't be there in a patient that doesn't have cancer. And what's good about that is that instead of biopsying a specific lesion, like let's say somebody has 10 different lesions spread through their body from the tumor, All of those might not be exactly the same. So the advantage of a liquid, what we call a liquid biopsy, it's not an actual biopsy, it's just a blood test, is that we're able to see what's circulating in the system. And so all 10 of those lesions are, excreting some DNA. And so how can we get a sense of what's going on, through the whole circulation? Obviously, it's not quite as, you're definitely on a needle in a haystack situation there. So to the tune of, 1 in a million. So it's not going to pick up every little thing, but it's going to pick up the dominant clones. And again, those were just recently approved. And so we're getting more and more data on those.
And it seems like we are on the precipice of some great scientific innovation. If you had to look in your crystal ball, where would you see this area moving in the next 5 to 10 years? Are we really going to be able to pinpoint that exact cancer and find a treatment for those patients? Or are we
I
still...
hope so. So I think that there's going to be increasing use of these liquid biopsies to find specific gene alterations. And if you think about the impact it could have on the field, one of the biggest impacts circulating tumor DNA might have, for instance, is not to treat people who don't need it. So when you have a colon cancer removed or a gastric cancer removed or something like that, we end up having to treat all of those patients with what's called adjuvant therapy. In other words, chemotherapy for 6 months or whatever it is to increase the cure rate. And it often adds 10 percentage points of cure rate. So nobody wants to leave that on the table. But the question is, aren't there some people who don't need it? Yeah, there are. In fact, a lot of the surgeries we do, over half the patients are cured with surgery alone. And if you start doing the math of, wow, wouldn't it be great if we didn't have to treat all those folks who don't need it? Problem is, we don't know who's who. If you see 10 people with exactly the same scenario, you might have 5 or 6 of them are destined to be cured with no further therapy. You don't know who they are. You're going to cure a couple people with therapy, and then a couple people are going to be resistant to it. So these circulating tumor DNA tests could be incredibly powerful to say to somebody, look, you don't have any cancer in your body. We're not picking anything up. We'll keep checking it. We're going to keep following you for 5 years, but we don't think you need to undergo therapy and take time off work and maybe lose your hair and have your white counts go down and have co-pays, all the stuff you think about it does to somebody's life to undergo chemotherapy. Wouldn't it be great if we could spare them that? Not to mention the costs, right? We can spend our money on COVID vaccines and not worry about cancer, right? So that would be a huge advance. The other thing is better therapies for these mutated genes. KRAS, for instance, is the most commonly mutated gene in all of cancer. It controls growth, not surprisingly. And of course, cancer, the hallmark of cancer is uncontrolled growth. So if we could figure out a way to specifically target that, it's a huge challenge, but people are rising to the challenge. And we now have drugs that are actually doing that. So I think the other big advance will be, treating people specifically based on the mutations they have. The third thing that's coming that also presents its own set of challenges is immunotherapy.
Mm.
And what's interesting about immunotherapy is now you're talking about an interaction. It's not just specifically, okay, here's a tumor and we're just going to bomb it, with chemotherapy or something like that. With immune therapy, you now have to take into account the patient's own immune system. And it turns out that we have very different, that the immune system is incredibly complex, not surprisingly. And there are lots and lots of versions of various genes that determine the functionality of our immune systems. And so we may have a day where not only are you testing the tumor itself to say, okay, there's mutations in gene X and gene Y and gene Z. You might also need to test the normal immune cell genes and say, okay, this person is a certain HLA haplotype, which means they can present on their antigen presenting cells a specific mutation. And this person can present this one and that person can present that one. So those two people, not based on the tumor, but actually based on their own immune system, might actually need slightly different therapies. And that is a level of complexity that I think will be coming. It's actually very difficult. That those immune system genes are incredibly variable, and the control is very tight on those things. And so eventually, I think we'll be needing to look, as immunotherapy gets more and more common, we'll need to be thinking about the patient's own immune system and how it interacts with the tumor, not just about the tumor itself. And that's going to require a lot of immunology expertise, which those of us in oncology, we have more and more of because we're increasingly doing immunotherapy. I do a lot of phase 1 studies. I've done over 20 first-in-human studies in my career, and all of them nowadays are immune therapies. Some of them are cell-based therapies. Others we have bispecific antibodies and sort of other ways to modulate the immune system. So that's required a lot of expertise on our part in terms of how do we handle these things. The side effects are totally different. You get sort of activation of the immune system. You don't get low blood counts. You don't get nausea. You get rash, you get fever, you get autoimmune diseases, totally different set of challenges that we have. And so, again, you need folks who are pretty well versed in that and know what's coming down the pike in terms of what's going to be the new immune therapies to hit the clinic in 2 years, 3 years. So all of that stuff requires, a lot of meetings, a lot of, presentations and things, just learning about how some of the top scientists in the world are thinking about immunotherapy for cancer.
Dr. Messersmith, I can't thank you enough for sharing your tremendous insights and your perspective on the future of this field. It's really been a delight speaking with you. Thank you so much.
Absolutely, it's my pleasure. Thank you so much.